All right. Thank you so much for the introduction and for the opportunity to share with everyone some of the latest innovations from the team at Vector Builder. Now, today we are going to spend quite a bit of time going through one of our new and very exciting offerings, MiniVec, our miniaturized plasmid. But before we dive into that, I do want to take a moment to highlight how Vector Builder offers end-to-end solutions for all areas of gene delivery. And when we're talking about the kind of scope, the perspective of what we at Vector Builder focus on, it's pretty all encompassing because when we talk about gene delivery, what we're talking about is any process where we take a foreign piece of genetic material and deliver it to a host cell. Now, this pretty simple definition is incredibly impactful for a wide array of different fields, including life sciences research and genetic medicine. And with the availability of new and optimized delivery methods and approaches, there's a huge spotlight, especially on the advancement of genetic medicine, with all of the opportunities and all of the potential avenues that exist there. However, of course, we all know that the pathway to the clinic is never easy. Now, while this pipeline for drug development can appear straightforward at first glance, of course, it is fraught with these roadblocks and obstacles. Even early on during research and development and early candidate selection, there are some of these pain points here that can sometimes fly under the radar. Our recent study analyzed plasmids that were created by lab and industry partners, or they were received from collaborators or from repositories. And we found that of these more than 2,500 plasmids, 50% of them contained either design or sequence errors. And when we look at these sequence errors, a good percentage of those were found through very straightforward restriction enzyme digestion, and then more were discovered through Sanger sequencing. Now, importantly, we did find a particularly high frequency of these sequence errors in AAV ITRs. And with such a high prevalence of these undetected inconsistencies, it's likely that some good candidates are just abandoned early on in the pipeline. But even after this, as we go through preclinical and then clinical stages, these pain points persist and arguably amplify with concerns like batch consistency and full, partial, and empty capsid ratios. All of these things, plus many, many more different pain points, many more obstacles, all gather together to contribute to a pretty astronomically high rate of failure of therapies in clinical trial. Currently, around 90% of these clinical trials fail due to largely lack of efficacy and toxicity. So, with 10% of these therapies making it to market, how do we streamline this entire process and give these therapies the best chance from day one? Now, at Vector Builder, we aim to smooth this pipeline from start all the way to finish. We work with both university researchers and industry partners to remove these pain points step by step all along this entire pipeline and make the pathway to clinic much more efficient. We've worked with tens of thousands of customers on projects ranging from vector design all the way through to clinical manufacturing. And our expert team has decades of collective experience in a range of areas of gene delivery, starting with vector design. Now, this is a huge component of what Vector Builder is, as our name implies. We currently help with the design of around 250,000 vectors per year, and we recently hit a really big milestone that we are incredibly proud of. There have now been more than 1 million vectors created on the Vector Builder platform. And further, we offer downstream plasmid preparation and virus packaging, totaling around 100,000 projects per year, as well as CRO services like library screening, stable cell line engineering, AAV capsid evolution, biodistribution assays, and more. And finally, we carry projects through the rest of the pipeline into GMP manufacturing, completing over 50 projects since the initiation of our CDMO services. Vector Builder, we're committed to helping you get from the bench to the bedside. And one of the most popular ways users start this journey is through the Vector Design Studio. Our innovative and intuitive tool allows users to select from over 120 different vector systems that are divided by desired application, including overexpression, knockdown, CRISPR, mRNA expression. And we also offer a wide variety of both viral and non-viral backbones, allowing you to have the ideal starting material. From here, you can plug in components from our vast database or paste your own sequences to create your ideal vector in minutes. And while this is an invaluable tool for many researchers, these vectors in the Design Studio are somewhat standardized with popular antibiotic selection markers like ampicillin. However, users can select other antibiotic selection markers like kanamycin, which is going to be more appropriate for those clinical applications. But we often see researchers hesitant to dive into full removal of those antibiotic resistance markers. Often utilizing these plasmids without any antibiotic selection, these miniaturized plasmids, it comes with some risks like lack of confidence in the validity or the safety, as well as compromises like trading antibiotic selection for supplement-based selection. However, at Vector Builder, we have not only developed our own miniaturized plasmid, MiniVec, with both antibiotic-free and supplement-free fermentation, but we've also heavily invested in the development of this offering to ensure its superior efficacy, safety, and manufacturability. So from here, I'm going to hand things over to our application scientist, Cole, who is going to walk you through all things MiniVec and really showcase how much we have devoted to this really exciting new offering.
Thank you, Connie. Hi all, this is Cole. So let me briefly introduce a few highlights of our mixed level miniaturized backbone. So first thing first, the backbone size is actually as short as 500 base pair. It has its own engineered or proprietary engineered E. coli strain that is called mini-host. So for this MiniVec, with such a small size backbone, we are observing a better efficacy, better safety, and better manufacturability. The most critical thing is actually this backbone feature the minimal level of bacterial origin, which normally in a regular plasmid, you will have kind of long origin of replication and also ampicillin resistant gene or a kanamycin resistant gene, for example. So in our design, the origin of replication is short. We also have another VEXAC B sequence that is used for a positive screening mechanism, which allows us to do antibiotic-free selection, positive selection of the correctly transformed mini-host. That being said, fermentation also does not require any special supplement, and we can still maintain this positive pressure so that ultimately we will get an increased plasmid use. So with this MiniVec and all these features together, we are also observing a better enhanced transgene expression, both in vitro and in vivo. We have been validating this MiniVec system in a few different applications, including non-viral regular overexpression plasmid, lentivirus, AAV, transposition systems such as piggyback, Sleeping Beauty, CRISPR-Cas locked-in system, and also many other applications. So before I actually dive deeper into each application, I think it's actually better to let me introduce the mechanism behind how we actually achieve such a miniaturized backbone while being like an antibiotic and supplement-free. So let's take a look here. So in our mini-host E. coli strain, we have engineered it with a growth inhibitor and a regulator. So right now, without MiniVec, this growth inhibitor that is right downstream of a VEXAC A sequence will get transcribed, and keep translating. And this growth inhibitor will actually stop the E. coli strain from proliferating. So what we need to do is to add supplement and trigger the translation of the regulator. And this regulator is going to help blocking the growth inhibitor. So ultimately, this mini-host E. coli strain can actually be proliferated like a regular E. coli strain. Then once we remove the supplement, mini-host cannot survive. So what we need to do is the transformation of the MiniVec plasmid into the mini-host. With the correct MiniVec inside the E. coli, this WAC-SEC-B sequence indicating here will be transcribed. This short sequence of RNA is going to hybridize with the mRNA that contains WAC-SEC-A upstream of the growth inhibitor. So I guess like some people can already tell that with WAC-SEC-A and WAC-SEC-B, they are actually a pair. So what is in our design is actually they are complementary. So once they hybridize, the double-strand RNA cannot be smoothly translated into the protein of the growth inhibitor. So this way, we don't need the supplement to express the regulator. And meanwhile, we can still block the translation of the growth inhibitor. The E. coli can be surviving and proliferating. So this is how we achieve the positive screening pressure. So of course, without the MiniVec inside the mini-host, there will be constant expression of the growth inhibitor that is gonna kill the E. coli that has lost, many copies of the MiniVec containing a mutated or like the deleted version of the MiniVec plasmid, or maybe without any MiniVec at all. So this is ultimately very good for MiniVec production and also large-scale fermentation, in which antibiotic is definitely, it cannot be used in large-scale clinical manufacturing. So in such system, maintaining high copy is actually very critical for a good plasmid yield. So now let me show you guys some data about the production of MiniVec plasmid first. So we have performed some strict fast lab scale production of the MiniVec plasmid. So what we observe here is that no resistant gene in the backbone. It actually means lowering the metabolic burden. The E. coli has more resource to actually replicate your MiniVec plasmid rather than transcribing and translating the resistant gene. Of course, also the smaller the size of the backbone, the smaller size of your entire plasmid. So that also means that you are going to spend your resources in the exact place where they are needed to have more of your gene of interest. And like I said just now, the continuous positive pressure is going to favor the high copy number survival of the positive mini-host. So as you can see in the graph here, we have been trying different MiniVec plasmid, including like a AAV transfer vector that includes like a ITR and franking the GOI. There's also lentivirus, the LTR franking GOI. Piggyback and Sleeping Beauty also having the ITR franking your gene of interest that is going to be integrated, and also regular plasmid that has an overexpression system on it. We are observing a much better molecular yield of this MiniVec plasmid compared to a traditional backbone. So now we know that the production is much better. Let's start using it in our daily experiment. So first, we want to demonstrate that it has a much better transfection efficiency and also overexpression function. So what we did here is to perform an equimolar fair comparison transient transfection into the HEC293 T cells. So as a result, we are observing a much better mean forens intensity that is 48 hours after the transfection into HEC293 T, and the positive rate is actually also higher. The M-cherry here is in equimolar as well. That is used as a control, and you can see that there is barely any difference in the M-cherry, while the EGFP, you can see a significant boost. So miniaturized backbone itself has been extensively reported for less gene silencing. The mechanism behind there are a few different theories behind it. The most major trend of explanation would be, the minimal level of prokaryotic sequence is gonna, like, usually the prokaryotic sequence will be packed into a very tight chromosome-like, chromatin-like structure, which is not actually optimal for your transcription and translation. So having your eukaryotic sequence, which is your promoter and your gene of interest, occupying most of the space in your plasmid, it will be constantly transcribing and translating, and the chromatin-like structure is more like a eukaryotic status. This can ultimately enhance the overexpression, and also, with a smaller size, it is definitely easier for transfection and also nuclear import when you want to express this kind of vector in your target cells. So now that we know that it gets better expression and it can transfer easily into the cells, what we want to do next is start integrating some genes in target cells, which is for some gene therapy, that will be the main purpose. So the first system we try is transposition system. We have tried two transposase. One is the piggyback, and the other is the sleeping beauty. So in this experiment, we actually use two vectors. One is a plasmid vector containing the transposase itself. Another one is the ITR franking EGFP gene of interest. So both of them are transfected in equimolar into the HEC293 T cells, and we keep passaging them. So in this experiment, both traditional plasmid of the two vector or both vector being replaced with the MiniVec plasmid were tried, and we are seeing from the passage 1 all the way to passage 9, there are a much better mean fluorescence intensity and positive rate. So this is again showing us that the expression of the transposase, and also, wherever we needed the MiniVec ITR GOI to be in your nucleus so that it can be integrated, they are there, and they are better than traditional plasmid design. And the next thing we try is a very popular system in recent years, that is the CRISPR-Cas system. So here we first demonstrated with non-homologous lock-in, because with the homology arms recombination, maybe it will be very application specific. So here, non-homologous recombination will be just fine. So what we did again is two plasmid vector. One is containing the Cas protein with a guide RNA sequence. The other one will be guide RNA franking the donor EGFP, I'm sorry. So with both plasmid replace my MiniVec, since day 2 up all the way up until day 19, we are seeing a higher positive rate and a higher fluorescence intensity. This higher fluorescence intensity show more successful non-homologous lock-in using the MiniVec backbone donor. Of course, we know that linearized plasmid sometimes can actually ensure a higher locked-in efficiency. So here we are just demonstrating a comparison in which a traditional plasmid is used, but what happens if you replace MiniVec? It shows that it is more accessible. It can actually enhance certain application where maybe linearized plasmid is actually when it cannot be used. So anyway, now that we have done some transposition and the CRISPR-Cas, the next thing that we always use for gene integration will be lentivirus. So lentivirus, the most flexible way of producing lentivirus is using a 4 plasmid system. So they include the gene of interest transfer plasmid, there is the GECPO plasmid, there is also the REF plasmid, and also the envelope plasmid, and here we use a VSVG pseudotype lentivirus, the most commonly used envelope, as a demonstration. So by replacing the backbone on 4 of this plasmid into MiniVec, we actually observe a much higher functional titer in the crude harvest. So we don't do any extra purification or any concentration that might actually interfere with the results. We use the crude harvest directly, in fact, to transduce the HEC293 T cells. We have been observing a much better functionality, potency of this lentivirus with a higher green fluorescence intensity and positive rate. So this again shows that, with the features brought by the MiniVec system, the virus packaging has been in favors. So okay, now that is enough for our in vitro experiment, and let's start doing something in vivo and see whether when we use it as a negative DNA in animal models, whether it shows some superior performance. So the first experiment we try is to do intramuscular and intravenous injection into the mouse model. So for our tailwing injection, which is the intravenous injection of luciferase 2 plasmid, we are seeing a higher bioluminescence in the liver region from day 1 to day 3. But of course, because when we do intravenous injection, most of the uptake of the plasmid will be accumulated in liver, which is known for a quick degradation of plasmid DNA. So anything post day 3 will be very minimal level observable bioluminescence signal. But then when we try to do intramuscular injection, which is one of the most common way to do a vaccine injection, we are seeing that the overexpression of the luciferase actually continues to be superior up until day 21 when compared to traditional plasmid. And the mouse model bioluminescence picture can be seen here. So our in vivo data continues to demonstrate a better performance of MiniVec even by directly delivering the negative DNA into animal models. So now we know that the performance, the potency is better when using your MiniVec plasmid. We need to prove that there is no extra additional toxicity effect that we brought to the application of using negative DNA. So first of all, we do an acute toxicity study. In this study, we do an intramuscular injection of the MiniVec plasmid that does not contain any meaningful GOI. We do a 400 microgram injection, single dose at day 0. So in both the male group and the female group, which the weight is around like 25 grams for the male and 20 grams for the female, we are not seeing any significant impact on the weight gain. Also, we are observing their behavior, diet, hair, and stool sample, and we don't observe any abnormal performance that is in both the negative control TBS group, the traditional plasmid, and MiniVec. And these are actually aligning with our expectation because plasmid DNA are going to be inducing less toxicity. But of course, acute toxicity is not enough. We want to do, how about repeated dosing on the mouse model? So then the next experiment we do is actually a repeated dose study that we actually start doing 2 groups. One is the low dose, so 10 microgram each time, and a high dose of 15 microgram of negative plasmid each time. We inject it twice a week, 7 weeks in total, intramuscularly into our mouse model. So overall, if you look at the body weight change, there is no obvious drawback by this continuous dosing of both traditional and MiniVec plasmid which is good. So when we compare it to negative control and transitional plasmid, as I was explaining just now, of course, we are not expecting to see any toxicity because it is already well known that plasmid DNA is not going to trigger a very obvious toxicity effect. But then, of course, we need to ensure it's really safe. So we also observe the clinical chemistry parameters and also the hematological parameters. They are all within the normal ranges. But then we also look at, the actual figures here from the ALT and AST. So these are the representative of any, severe liver damage. So from the result that we are seeing here, they are all within the normal ranges. And it demonstrated that we are not seeing any obvious significant liver damage, as well as we check the creatinine level, the urea, and all other parameters. They are also not showing any kidney damage as well, which is very important for toxicity. So, okay, now that we know that it's not toxic, we need to start using it in a case study and show that, MiniVec backbone is actually a better candidate for any future negative DNA, pipeline or any drug design. So here we use a very recent vaccine as an example, which is the COVID-19 protein vaccine. So this MiniVec is simply with a promoter upstream of COVID-19 spike protein. And our dosing is for 3 times. So the first jab and then 2 booster jab. So this experiment, in each dose of the immunization, we are putting roughly 100 microgram, which is 1.4 times 10 to the 13th copies of plasmid at day 0, 14, 28. So that is every other 2 weeks. So in this experiment, the immunization is done intramuscularly, and also serum sample will be collected 2 weeks after the immunization, which is right before the subsequent immunization. So this study is actually still ongoing. So I'm showing some preliminary data here that in the first dose and second dose, we are seeing a much better spike protein specific antibodies, IgG, that is being elicited by the MiniVec backbone compared to the traditional plasmid backbone. And as we were demonstrating earlier, maybe first thing is a better efficiency of getting into cells and also a better overexpression, which can actually help us, generating a better immune response. But of course, this is not enough. We are also performing extra tests such as harvesting the splenocyte from the mouse model and perform interferon gamma secreting cells ELISpot after we culture the splenocytes. So I'm sorry, this one, we don't have comparison with the traditional plasmid yet, which we are, as I was saying, the experiment is still ongoing, but we have comparing it to a MiniVec backbone that has empty cargo on it. So here we are seeing that this spike protein specific interferon gamma secreting cells is actually pretty good after we stimulate the splenocyte with spike protein, recombinant spike protein. And meanwhile, when we culture the splenocytes, it constantly secretes a high level of interferon gamma and interleukin 2. So these are all indicating that MiniVec backbone is actually a very good potential helper for your negative DNA design for any future application that you may consider to use negative DNA delivery for cell and gene therapy. Okay, so that is all the data for my MiniVec validation experiment. So here I will offer 3 take-home messages for the MiniVec. First thing, it gives us a very good expression efficacy, and it ensures a high level of transfection efficiency. So that is already the first good thing. And the second good thing is because of the theory that we just talked about, less gene silencing, and also, a better level of the expression in vitro and in vivo are observed in our experiment. And the second thing is the safety aspect of using MiniVec, because it contains the minimized level of the backbone. That also means a minimized level of prokaryotic sequence, the origin, and we don't even have the antibiotic resistant gene. So by relying on an RNA hybridization effect that is done by our vaccine A and B, our E. coli can be continuously positive screened and it can proliferate. And in this entire process, we don't need any special supplement, which is very important that we are not introducing extra redundant materials during the manufacturing process, which we might need to justify to the regulatory bodies, like why we are adding this just for the yield. So there's no concern of this anymore. So the third thing is about manufacturability. So our MiniVec has been demonstrating much higher plasmid yield compared to traditional backbone. And meanwhile, lentiviral vectors as one of the hottest viral vector that is used in cell therapy these days, like CAR-T. The production cost is actually very high. The turnaround time could be high. So leveraging the features of MiniVec, we trust that the production of lentivirus will be better by minimizing the cost and also the turnaround time for the manufacturing development work. Basically, that's it. Thank you, all of you. So if there's any question, feel free to ask us. We are here to address your questions. Thank you all.